Hello everyone, let's explore how generative and agentic AI will reinvent life sciences discovery. For most of modern medicine, discovery has looked like this. A scientist reads the literature, forms a hypothesis, designs an experiment, runs it, interprets results, and repeats. It is powerful, but it is slow. It is also fragile. Results are scattered across papers, lab notebooks, slide decks, and private datasets. Decisions are made in meetings. The process depends on coordination across biology, chemistry, data science, clinical insight, and operations. And the painful truth is that even when we do everything right, biology is still the ultimate bottleneck. Now imagine a different operating model. You start with a goal like find new targets for Parkinson's or identify a therapeutic approach for a rare disease with limited prior research. Instead of a single model producing a list of suggestions, you have an AI team, multiple specialized agents, each with a defined role, working together across data sources and tools, planning multi-step investigations, executing analyses, proposing experiments, and iterating, not as a replacement for scientists, but as a new layer of automation that turns the discovery cycle into something closer to continuous engineering. That is the shift from predictive AI to generative AI to agentic AI. Predictive AI gave us better property prediction. Generative AI expanded the design space, making it practical to propose new molecules. But both of those waves are still mostly passive. Humans still orchestrate the workflow, move files around, interpret outputs, and decide what to do next. The emerging wave is agentic systems that can reason, act, observe, and reflect in loops using tools, memory, and external data to carry out complicated work end to end. In drug discovery terms, this matters because the pipeline is not one problem. It is a chain of interdependent problems. Target identification and validation, hit discovery, lead optimization through iterative design, make, test, analyze cycles, preclinical safety, and eventually clinical development. Each stage produces different data types and different forms of evidence. Omics, imaging, assays, pharmacokinetics, clinical outcomes. And across the whole chain, a major bottleneck is not just experiments, it is decision making, searching, collating, interpreting, and communicating evidence across teams. Agentic AI is being positioned as a way to compress that decision overhead and connect the steps into traceable, reproducible workflows. In the agentic framing, the model is not the product. The workflow is the product. To understand where this is going, it helps to picture discovery as a loop. You design a hypothesis, you test it, you analyze results, you refine. Traditional discovery runs this loop slowly and sequentially, constrained by people, working hours, coordination friction, and limited throughput. The AI-driven vision is a continuous loop system that can run many iterations, generate large audit-ready datasets, and keep a persistent memory of what worked, what failed, and why. Some of that future is already visible today. We now have AI-designed molecules in human trials. The milestone is not just that models can propose molecules. It is that programs built with AI platforms have progressed into mid-stage development, forcing the industry to ask a sharper question. Is it simply faster or is it better? One framing that is starting to resonate is that success should not be measured only by speed or return on investment, but by return on experiment, meaning how much validated biological insight each cycle produces per unit, time, and cost. Because in the end, biology does not care how elegant your model or solution is. Translation to humans is still the test. So what exactly changes when generative AI becomes agentic and agentic becomes embodied inside discovery platforms? First, literature and knowledge work stops being a human tax. A specialized knowledge agent can continuously read and synthesize unstructured biomedical text, clinical data, and even modalities like medical imaging, extracting findings, building cohorts, and producing structured summaries with provenance. In the platform view, this is no longer a one-off search. It is a living knowledge layer that is versioned, traceable, and tied directly to decision points. That matters because the biggest hidden cost in R&D is duplicated thinking. 10 teams reading the same papers, 10 versions of the same summary, 10 meetings to align on what is known. Agents can reduce that friction, but only if they operate with citations and data lineage. Second, target discovery becomes more multimodal and more systematic. Instead of relying on one dataset or one hypothesis, an agentic system can integrate multiple signals, genomic and proteomic data, phenotypic results, pathway models, and real-world evidence, then propose ranked targets with supporting evidence, counterarguments, and suggested experiments to validate. A human still chooses, but the search space gets wider, and the reasoning becomes more explicit. Third, the design space opens further, but with more constraints. Generative models are good at proposing molecules, but discovery is not a molecule beauty contest. The molecule has to be potent, selective, safe, manufacturable, and viable in the real world. Reviews of computational drug design emphasize how representation choices and evaluation metrics shape what good looks like. Linear representations like smiles are efficient but lose spatial context. Two-dimensional graphs preserve connectivity. Three-dimensional representations have higher biological fidelity but cost more computationally. Different generative approaches like encoder-decoder methods, GANs, reinforcement learning systems, and diffusion models come with different trade-offs. The point is not that one model wins. The point is that an agentic workflow can combine them, using the right representation and the right tool at each step, rather than forcing everything through one model. Fourth, property prediction and risk triage become continuous, not episodic. A preclinical analyst agent can run in silico simulations and predict properties like potency, permeability, pharmacokinetics, and toxicity. An orchestrator can then triage candidates into promote, archive, or optimize, and feed optimization back into generation. This is the heart of the rapid in silico loop. The promise is not that you eliminate wet lab testing. The promise is that you move more failure earlier, and you spend scarce lab capacity on the candidates with the highest probability of success. Fifth, the make step starts to change character. Robotic labs can run around the clock and produce standardized, reproducible data. When agentic planning systems can generate protocols, schedule experiments, interface with lab automation, and capture results into structured data stores, the loop tightens. The workflow becomes like a self-improving production system for evidence. If that sounds abstract, here is a concrete way to think about it. A modern agentic discovery platform can be described as a team of agents. You have an orchestrator agent. Its job is to interpret the goal, break the project into tasks, sequence actions, evaluate results, and adapt the plan. It is the project manager and systems engineer. You have a knowledge agent. Its job is to search and synthesize literature and internal corpora, extract key facts, map relationships, and keep citations and provenance. You have a molecular architect. Its job is to produce or retrieve target structures, run docking or structural simulation, and provide the structural blueprint for design. You have a preclinical analyst. Its job is to predict ADMET and functional properties, rank candidates, and flag risks. And in more advanced systems, you can add a synthesis planning agent, an assay design agent, a toxicology specialist agent, a regulatory documentation agent, and a portfolio strategy agent. The deeper innovation is not any single agent, it is the modularity. A platform can swap models in and out, attach new tools, and run workflows with traceable configuration. This is how discovery becomes scalable. Teams stop operating as isolated experts and start operating as users of a shared workflow system. This is also where infrastructure becomes a differentiator, not just compute, but governance. The industry is learning that AI must be treated like a scientific reagent. It needs versioning, lifecycle management, drift monitoring, and traceable retraining conditions. Regulators are moving toward principles like transparency of model lineage and data provenance, assessment of bias in clinical context, and validation plans that connect in silico predictions to measurable experimental outcomes. Trustworthy AI in pharmacology becomes less about flashy demos and more about discipline. Prospective hypotheses that are locked and tested, bridges that connect model outputs to assayable biology, model lifecycle governance, and a clear human in the loop, not as a rubber stamp, but as accountable scientific leadership. A useful mental model here is the centaur. Not the idea that AI does everything, but that the best outcomes come from combining algorithmic speed with human judgment. Many leading platforms in the field have embraced this in practice, integrating AI into the design and analysis loop while keeping expert scientists responsible for key decisions. That balance matters because the hardest part of discovery is not generating ideas. It is choosing which ideas to kill. And humans remain uniquely good at integrating messy context, organizational priorities, and the lived reality of biology. Now, let's talk about the future a little more boldly. Over the next decade, the life sciences will likely shift from tools to operating systems. In the tool era, you buy a docking tool, a QSAR model, a generative chemistry model, a literature search system, and a data warehouse. You glue them together with scripts and meetings. In the operating system era, the default interface becomes a conversational and programmable layer that can orchestrate those components in a governed way. A scientist asks a question. The system retrieves internal and external knowledge with citations. It proposes a plan. It runs analyses. It logs decisions. It produces a report with full provenance. It can hand off to a robotic lab or generate a protocol ready for execution. It learns from outcomes and improves future proposals. Several innovation patterns are already visible across the ecosystem. One pattern is the rise of AI-native workflows designed to tackle fragmentation with an emphasis on transparency, reproducibility, and scalability. The idea is that discovery is not just a model problem, it is a workflow and traceability problem. Another pattern is multi-agent architectures that mimic real pharmaceutical teams, where roles like target discovery, lead identification, lead optimization, and preclinical evaluation are simulated as collaborating agents, each equipped with domain tools. Another pattern is cloud-centric reference architectures for agentic workflows, where retrieval systems support literature synthesis with citations, training infrastructure supports large-scale simulations, and agent builders allow modular composition of workflows. Another pattern is the recognition that model management is becoming a first-class need. As the number of models grows, with different inputs and outputs, different versions, and different validation states, the organization needs a model registry and execution layer that can handle asynchronous execution, scaling, and consensus across multiple computational models. Without this, reproducibility collapses and operational friction returns. If you zoom out, you can see the shape of the endgame. It is not one AI model discovering drugs. It is a network of AI services and agents tied into a governed enterprise platform, connected to data pipelines and lab automation and supervised by accountable humans. In that world, what does discovery feel like day to day? It feels less like hunting for answers and more like steering a system. A disease area lead can ask for an updated target landscape and get a ranked list with evidence and uncertainty. A chemist can request 10 optimized series with constraints on potency, selectivity, and synthetic feasibility, and get proposals along with the rationale. A safety lead can request early toxicity flags and get hypotheses and suggested assays. A project team can run hundreds or thousands of design loops in silico and reserve wet lab cycles for the most complex tests. And crucially, the system keeps score. It tracks what it proposed, what was tested, what failed, what succeeded, and how reality differed from prediction. That learning loop is how platforms get better. But the future is not automatically bright. There are real risks and hard constraints. Data heterogeneity and data quality remain fundamental. AI is only as powerful as the data it consumes. And in pharma, data is often siloed, poorly modeled, and inaccessible. Without meticulous data capture and curation, you cannot get trustworthy outputs. Reliability is another constraint. Agents can hallucinate, misinterpret, or overgeneralize. In drug discovery, that can waste months or lead teams in the wrong direction. This is why guardrails, citations, and validation loops are not optional. Privacy and intellectual property are constraints. The value in pharma is in proprietary data and knowledge. Systems must support access control, audit trails, and safe use of sensitive corpora. And then there is the biggest constraint of all, biology. AI can accelerate hypothesis generation, but it cannot eliminate the complexity of human physiology. Many promising ideas fail in the clinic. Faster iteration may mean faster failures. That is not a bug. It is a feature if you treat it as learning. The goal is not to avoid failure. The goal is to fail earlier, cheaper, and more informatively, while increasing the probability that the survivors translate. So what should leaders, researchers, and investors watch for as signals that the agentic future is real, not hype? Watch for workflows that are closed loop, not just generative. A system that proposes molecules is interesting. A system that proposes, predicts, triages, optimizes, and logs the full chain is transformative. Watch for traceability. If you cannot answer which model version, which data sources, and which prompts produced an output, you do not have an enterprise discovery system. You have a prototype. Watch for integration with labs and data capture. The fastest way to kill AI value is to generate outputs that cannot be tested quickly or cannot be captured cleanly. Watch for governance. The organizations that treat models like regulated reagents with lifecycle management and validation plans will move faster long-term because they will trust their system. Watch for human AI collaboration that is designed, not accidental. The Centaur approach is not a slogan. It is a workflow design principle. Humans should be responsible for framing goals, defining target product profiles, choosing trade-offs, and making go or no-go decisions. Agents should take on search, synthesis, exploration, documentation, and routine planning. Now let's land this with a vivid picture of the next decade. In the near term, agentic AI will first dominate the knowledge layer. Literature synthesis, internal document retrieval, protocol drafting, and decision support will become faster and more standardized. Teams will use agents to reduce the overhead of alignment and documentation and to make evidence more searchable. In the next phase, agentic systems will take a bigger role in silico DMTA, design, make, test, analyze loops. They will coordinate generative chemistry with docking, property prediction, and multi-parameter optimization, producing narrower and higher quality candidate sets for synthesis. This is where cycle time compresses dramatically, not because the wet lab disappears, but because the lab becomes the validator rather than the explorer. In the next phase, the self-driving lab becomes practical for more organizations, not necessarily a fully autonomous facility, but a hybrid model where robots execute standardized protocols, agents schedule and interpret, and humans supervise and design the bigger strategy. The lab becomes a data engine. In the longer term, the most powerful change may be the emergence of discovery as a platform business. If you can reuse trained models, pipelines, and data sets across many programs, you get operating leverage. You can redistribute risk through milestone-based partnerships. You can build an engine that produces validated assets, not just software outputs. And as that happens, the competitive advantage shifts. It will not be just algorithm novelty. It will be integration depth, data quality, validation credibility, and the ability to translate computational insight into real clinical assets. So when people ask, what is the future of life sciences research and discovery with generative and agentic AI? The best answer is that it becomes more like continuous, traceable, high-throughput learning. Instead of science being limited by the speed of reading, meeting, and manual iteration, it is augmented by systems that can explore larger spaces, run more cycles, capture more evidence, and keep a disciplined memory of what the organization has learned. The winners will not be the teams with the flashiest demos. They will be the teams that build trustworthy workflows. Workflows that connect models to experiments, experiments to data, data to decisions, and decisions to outcomes with clear provenance at every step. In other words, the future is not that AI discovers drugs for us. The future is that we build self-improving discovery systems and scientists become the leaders who steer them. Thank you for watching.